So yes, this is chapter 9, cell communication. This chapter, I like how, especially this book, the other book didn't do this, but cell communication relates to the cell cycle, which is mitosis, which then also relates to reproduction, which is meiosis. So they're all very, very interrelated. And so the cell communication basically determines when the cell is going to go through the cell cycle, or through mitosis. A couple of vocabulary terms you need to know. First off, the ligand. The ligand is what actually is contacting. So if you reach out and you touch someone, that's your ligand, okay? It's actually the signal molecule, okay? You reach out and you flick someone in the nose, that's a ligand. That flick is a ligand, okay? It's a signal. Your nose is the receptor, okay? And then that pain signal that goes through is in that what's called a signal transduction pathway. So transduce means to create. So it's creating this pathway as the signal is being sent by the ligand. Okay? And you notice that there are two different types of ligands. There are those that are hydrophilic, which access on the outside of the cell membrane. Those that are hydrophobic, which access the receptor molecule on the inside within the cytoplasm. It's hydrophilic is on the outside, hydrophobic on the inside. And then the reason is because, again, this is that biphospholipid layer. So the hydrophobic is able to transfer through, whereas the hydrophilic would not be able to. And again, it's this basic process. The ligand attaches, it stimulates a response. That response goes through a signal transduction pathway, which then gives a response. Okay. So you think back to behavior. You think back to our animal behavior unit. Behavior, all the behavior is just a learned response over time. So essentially, a signal transduction pathway is just a response, but in a short amount of time. And this could be something as much as you touching a hot stove and pulling your hand back, to the point of you have too much sugar. Like after Halloween, you'll have so much sugar, the insulin in your body is going to start breaking down that sugar. Okay, that would be a signal transduction pathway. If you have too much or you have too little, that will then release these kind of signals. There's different types of signals. There's different ways of the signal pathway. Okay. If you just go out and you're touching on cell, okay, it's direct contact. Just like it's called, direct contact. It's through these gap junctions. So if you look up here, each cell can be very, very closely together. Now, if this is animal cells, it's very easy because animal cells, again, they don't have a cell wall, and so they're going to connect very easily through that plasma membrane. But if it's an animal cell, they've got something called plasmodesmata. I'll spell that for you. So again, if it's an animal cell, okay, because an animal cell, it's got the cell membrane, and it's got the cell wall. That's going to be connected to another cell wall, the cell membrane. There's going to be little junctions right here. There's going to be gaps in the hole, in the wall. So those are called plasmodesmata. So PLASMODESMATA. So those little connections right here, this is the plasmodesmata. Think if I had a girl, I'd call her plasmodesmata. It's a good, like, Hispanic name, right? I think so. I don't know, it sounds like a, okay, I'm going to stop now. What? Oh, it sounds like Antonio Banderas. You all see, like, that movie he had with the little guitar case that he, like, shot rockets out of? Desperado? That's what it was. They call me plasmodesmata. Okay. So that connection is the direct contact. That direct connect between the gap junctions. The next one is paracrine signaling. Okay, if we're developing, if we're an embryo, okay, that embryo, that one egg turns into two cells, and that one fertilized egg turns into a cell, an embryo. That embryo turns into two cells. That two cells turns into four, and it goes from there. Okay, and it's non-differentiated. It means it's not specifically a liver cell, or an eye cell, or a tooth cell, or whatever it's going to be. So what it can do is, let's say that this cell decides it's going to be an eye cell. Well, you don't want a bunch of eye cells growing next to a bunch of toe cells, because you need to have toes sticking out your eyes. It look really weird, right? It's cool and all because you can, like, run on your face, but you want all the cells around you to be exactly the same. So it will use what's called paracrine signaling. Okay? It affects all the cells locally around you. Okay? And the best way I've learned about this kind of description is that if I got a stack of sticky notes, okay, I can sit there and I can write, like if it's Chaz and Tristan, they can sit there and write notes back and forth to each other. If they wanted to, I guess. Okay? On those sticky notes. Very easily. Okay? Very easily they could connect, discuss between each other very, very easily. But they've got this stack of sticky notes and they want to share these notes around themselves. Very easily they could turn around from their chair, pull a sticky note off, and put it next to them or behind them or whatever they need to do. Okay? That's what's called paracrine signaling. It's a local signaling. One cell will send out a bunch of signals within that cell. Okay? That make sense? So direct contact is where it's actually touching. That paracrine signaling is where it can basically reach out and touch other cells, but it can't go very far. It's very, very local signaling, but it can touch a lot of cells. Okay? If this was a cell that's going through apoptosis. Apoptosis is the natural breakdown of cells. Okay? And your cell is going to slowly start breaking down. It's going to break down all the cells around it as well, especially if it's like a necrotic type cell. And so it's going to do paracrine signaling because it's breaking down other cells locally around it. Now I really should have put synaptic signaling next to paracrine because direct, paracrine, and synaptic are very locally signals. Okay? If it's direct, it's touching. If it's paracrine, it's very close. Synaptic, I'm going to go down to the very bottom. Synaptic signaling is where it's a very specific paracrine type signaling where it uses chemical signals called neurotransmitters. Okay? And if you look right here, again, if you remember the whole like hand, I've shown you that, right? About the nerve cell, how your fingers are the dendrites, this is the cell body, your hand is the cell body, and the arm is the axon. Okay? So if you've got, have I shown that?
You haven't showed us, but Miss Price did.
Okay.
Or Miss 80 or something.
So it's this idea that your nerve cells, okay, your nerve cells are basically made up of dendrites, and dendrites connect to the cell body. That's where the nucleus is, and that dendrite from that cell body then has a long axon. So always remember it as a hand. Okay, your fingers are your dendrites. That's going to send out the signals. Like you can jazz hands. Okay, so you jazz hands. So your cells are going to jazz hands their signal out. Okay, they're going to send out neurotransmitters. That's coming from the cell body. Those jazz hands are going to talk to the axon. So you talk like this. Now notice, do my fingers touch the elbow? No. That's what we call the synaptic gap, or the synapse. And these neurotransmitters have to go through that synapse. Okay? It's going to send this. That was really horrible back. It's going to send this this neurotransmitter through the synaptic gap. It's going to have to send these signals. These ligands are going to have to send out past one axon to the next dendrite. Okay? And it speaks axon to dendrite. Notice that the dendrites can grow out. And you can actually watch dendrites. There's really cool YouTube videos of dendrites growing. Okay? And you see them, they almost look like little bits of electricity they're shooting out. And that's actually what happens whenever you learn something, your dendrites actually make connections. Okay? So when you're learning, your dendrites are growing. And as your dendrites are growing, they're making more connections to other axons. And then you can make those connections. And so you truly are making like physical connections through these other transmitters. Okay? And if you look, this right here, the little blue part, okay, the little blue part right there, that's the receptors. These little reds, these are the neurotransmitters. The neurotransmitters are the ligands. Okay, it's going to cross the synaptic gap, or the synapse. Okay? So have y'all ever seen the Abilify commercial? Abilify commercial, yeah. It's an antidepressant.
Days I can put on a brave face and muddle through, but other days I still struggled with my depression. I was managing, but it always seemed to be creeping up on me. I felt stuck. I just couldn't shake my depression. So I talked to my doctor. He said adding Abilify to my antidepressant could help with my depression, and that some people had symptom improvement as early as 1 to 2 weeks. He also told me about a free trial offer from Abilify. Now I feel more in control of my depression.
Abilify is not for everyone. Call your doctor if your depression worsens or if you have unusual changes in behavior or thoughts of suicide. Antidepressants can increase
Not the one I'm looking for.
teens and young adults.
There's one, what is it? Oh, what's that? So here's what I was looking for, okay? Cymbalta, it talks about this whole idea where basically you've got these axons, okay? And these axons are going to send out these neurotransmitters. That's my axon, don't make fun of it. Right? These neurotransmitters are going to send out, okay? And this entire commercial is talking about this whole idea where these axons, these neurotransmitters, are going to come and talk to another signal cell, okay? And what it's saying is that this antidepressant, what happens is that it says that when you're depressed, based on what they're showing us, is that these neurotransmitters want to come over and connect to this response. Okay, there's this responding protein. Okay, and so again, these are the neurotransmitters. But what it's showing you in the commercial is how these neurotransmitters are bouncing back, which is causing the depression. Okay, and so what it does is it says basically, and I'm pretty sure it's Cymbalta, it comes and it's actually able to open up these receptor sites, and so to where these neurotransmitters will make the connection. Okay, to then have that signal and transduction response. It's not Cymbalta. It was a little one with a little like something. It's like it looks like a little blob. He's like bouncing along. He's all sad, and it goes into an animated form of neurotransmitter. It's really awesome. I need to find it.
I don't think that's Cymbalta. Because Cymbalta is that little butterfly.
So again, this is the endocrine system. This would be a much distant one, okay? If you are in danger, what is your natural response? You get two responses. What are they? Cry. No. Sit there and crawl up in a ball. Fight or flight, okay? So you have this fight or flight. You have an adrenal gland that's just above your kidneys, okay? That adrenal gland is not near your legs. But what it can do is it can produce adrenaline, and that adrenaline will then release into the blood system, and through the endocrine glands, release into the blood system, go to your muscles. Those muscles will then start producing more ATPs to where you can start running faster. Okay, your brain becomes much more sharp when you're in that fight or flight mechanism. But again, it's a distant translation. It's a distant signaling. Okay, so back to that sticky note idea. Okay, we can do direct contact where you just kind of write notes next to each other. You can do paracrine signaling where you actually hand the sticky notes out around you. Okay, but again, we're in the middle of the classroom, but people are in the far distant corners. Can they get paracrine signaling? No. So what they're going to have to do is they're going to have to use something else. They're going to have to use like a text message or a tweet to each other. Okay, and that text message and tweet, that can then be sent much further distances. That's like endocrine signaling. Okay, does that make sense? Okay, so those 4 different types. Those are the main 4 different types of cell signaling. It's all based on how locally you want to contact. Okay, now, these are kind of broke down. The direct connect and the paracrine, they both are used frequently in early cell development. So again, just like that eye cell, you want every cell next to that eye as an eye cell. You don't want any other kind of random cells over there. So the direct connect, that direct contact will actually connect both cells together and make sure they're on the same track. Same thing with paracrine signalings. This also is immune response. So if you get, let's say, an allergy, okay? Allergies are paracrine signaling. You get one little piece of pollen, let's say you're allergic to dust. You get one little piece of dust, it's going to cause the rest of your nose just to start like draining every single orifice out of your body, right? That immune response is because it's going to send that signal out to multiple locations. Whereas the endocrine system uses the circulatory system, it's both in plants and animals. Okay? Hormones. If y'all ever go to the Parker County Peach Festival, you know what I'm talking about? I want to say in late spring, there's a Parker County Peach Festival, and you can go buy peaches. Okay, but sometimes when you buy peaches, they're not ripe, and so what can you do with a peach? You just deal with it, right? Do what? You sit in the sun, and then most time what people do, or especially their tomatoes or whatever, they're not ripe enough, they'll put them in brown paper bags. Okay? The reason why is because of this paracrine signaling. That paracrine signaling is actually sending out what's called ethylene, and ethylene is a natural ripener. And so if you have one apple, or that one banana that's nasty in your drawer, okay, you ever notice that? You take one banana, and you have like one banana left, it's starting to turn a little brown and spotty, so you're like, oh, I can go to the store and get me some new bananas. And you leave those new bananas next to the old bananas. You ever heard like the one bad apple rumors a whole bunch, or one bad banana rumors. That's what it is. It's because what it's doing is doing this paragon signaling. It's sending out molecules to affect the closed cells. So it's ripening the other bananas faster than it should. So if you ever go buy new bananas, you always want to set them further away from the old banana, okay? Because of the paragon signaling. And then this neurotransmitter is very, very brief, very rapid. Okay? That's the reason why you can send signals. You touch a hot skillet, you're going to pull your hand back very, very quickly. Okay? Because it's very brief, very sudden. There's three main different types. There's the chemical gated channels, the enzymatic receptors, and G protein coupled. You need to know basically what they, which ones are which. Like you need to know, okay, what are three different types? The chemical gated, the enzymatic, and the G protein. Okay. Do you need to know what actually is going on? You basically need to know essentially that there is on the chemical gated, you do need to understand that a ligand attaches to the receptor cell that then opens up a gate. Okay. That's just like our active transport, whereas the gated channel, it's the same kind of idea. So a ligand attaches, opens up the gate, the ions can flow through. Whereas an enzymatic receptor, the ligand attaches, that then activates this enzyme that then creates a cellular response. Then the final is called a G protein coupled, and it's coupled because if one thing occurs, it causes another thing to occur, which causes another thing to occur. Okay? And G protein is just a specific protein that's in the plasma membrane, and that G protein will be affected by the ligand. The G protein will be affected by the ligand, that's the signal, that will then change this structure, which will then open up this ion channel, which will then activate this enzyme. So it's a coupled of reactions. It's this reaction causes this reaction, causes this reaction, which causes this reaction. Okay? So the chemical gated channels and the enzymatic receptors are very, very localized. The G protein is very, very long distance. It's because it's causing one to cause another to cause another. So again, that signal transduction pathway is basically made up of three parts. The ligand is received. Again, if it's hydrophilic, it's going to be on the cell membrane. If it's hydrophobic, it's going to be within the cell. It sets off a series of steps. Now, I've got examples of each of these steps on the later slides. And then it finally phosphorylates. And the phosphorylation, the kinases within that phosphorylation, and what does the phosphorylation mean? Stop there. What's it doing? Well, what do you hear in there? You hear phosphate. So it's either going to do what with the phosphate? It's either get rid of it or add to it. Okay. So phosphorylation, either you're going to remove it, and it's going to inactivate it, or you're going to add to it, it's going to activate it. Okay? So it all comes down to essentially these two molecules. Either you're going to phosphorylate or dephosphorylate. And if you're phosphorylating, you're going to use something that's called a kinase. And a kinase is actually where you're going to add the phosphate. But if you're going to take that phosphate away, you're going to use phosphatase. And these two enzymes both affect that. Okay. So basically what you need to know is that the protein kinases and the phosphatases allow for these responses, I'll come back to it, allow for each of these steps to occur. Okay, the feedback inhibition, the amplification, or the intermediates. What's feedback inhibition?
This is where the protein shuts down the first.
There you go. Yeah, the protein either shuts down if it's inhibition, or if it's activation, it allows for it, right? From the last part, the last product, it will then shut down the first enzyme. What does it mean to amplify? Do it? To get louder. Now, in the process of proteins, we're not going to make it louder. We're going to basically just make more responses. Okay? And then intermediate, what's an intermediate?
Chemistry?
Yeah, what is it in chemistry?
It's a product that becomes part of the reaction.
And that's exactly what it is in biology. If your goal is to make product D, an intermediate would be, it says it has to make product B to make product C to eventually make product D. It's the same idea. Okay, so it's those three ideas. So again, all that is because of this protein kinase. It's the actual phosphorylation. So the first one's feedback regulation. The example I gave you is gene expression. So the one they gave you in the book is regulation of gene expression. So hormones can actually regulate what kind of things are being transcribed. And what's gene transcription? You remember that from freshman biology? We haven't covered it yet. What are we doing? We're reading genes, okay? And where's the gene located? On the what? It's in the nucleus, but it's on this whole genome itself, which is DNA, okay? Gene transcription is taking that DNA and making what? RNA, okay? So what feedback regulation can do is if we have enough of these hormones will actually bind within the cell to basically block this transcription, okay? So it activates an inhibitor. It sounds backwards, but it can, and where it actually will block this hormone that binds the receptor, right, which will then stop the process of gene transcription.
Why would you want to do that?
Sometimes you don't want to go because you're going through and you're doing gene transcription several times. If you are, let's say you're going through cell growth, okay, but you need to regulate cell growth, and so if you're not producing enough growth hormones or you're producing too many, then this will not be blocked down, okay. But if you need to regulate how quickly or how slowly you grow, let's say like right now, as a developing adult, you probably have lots of growth hormone going through your body, right? And so you're slowly but surely developing. But once you hit about my age, I'm not going to develop anymore, right? If not younger than this. And so this is when this will come along. This feedback regulation, this hormone will basically slow all this down and slow the production of RNA. Yeah. And it's also if you just want to change your gene expression. So if let's say you just weaned your child, okay, which basically means no longer are you producing milk for your child. That child will stop producing normally, a normal animal, not so much humans, but before we got the mutation of the ability to break down lactose after being weaned, this feedback recognition would start going on. So you're weaned, your body would start producing these hormones that would then stop the production of the lactase enzyme. And so if you stop the production of lactase enzyme, you can no longer break down lactose. So we're not supposed to be able to drink milk. Okay? After we wean, after we break away from our mother, we're not supposed to drink milk. We're not supposed to be able to. So if you are lactose intolerant, you're actually normal. If you're not lactose intolerant, you're actually a mutant. Okay? So naturally, you're supposed to be able to go through this feedback recognition. It will then shut down the production of lactase because it's one of those things you just didn't need.
What a lame superpower.
I know. It's not flying or anything.
What about that whole thing with
Oh, you can't drink milk? I can.
14 or
You do, but you can find other sources of calcium. Broccoli is a good source of calcium. You eat 4 pounds of broccoli, but you probably don't want to do that.
Okay, well, not 4 pounds.
It's also high quality fiber. The next one is signal amplification. And so instead of getting louder, it's just making more and more. Okay? So a signal comes in, the protein receives the signal that then activates. And as it activates, and it's really weird, like the way they name this, I find it really very strange. I don't know if y'all can see it, but it says MKKK. And this one says MKK. And this one says MK. And then eventually you have the response, and then eventually you have the full cellular response. Okay? The reason why is because it's called mitigan activated protein, or MAP kinase cascade. It's a really fun word to say. But essentially what it means is that this mitigan activated protein, or this MAP kinase, will then activate the next one, which will then activate the next one, which will then have a response protein. Okay, so it's a step by step by step. But instead of just activating one protein from this one MK, it activates multiple proteins. Okay, and it names itself off of the final phosphorylation. It names itself off of the final activation. So again, if you notice, it was the phosphorylation which is causing all this to occur. So there's more phosphates there. Okay. So it does have that final response protein and then that then cellular response. So it's just like if I tell one person, and then one person tells two, and those two people then tell two more, and then two people, that then causes the amplification. Okay? Y'all know very quickly in high school, especially in the cafeteria, I don't know if y'all do so much, but here in the freshman campus, very quickly, rumors get spread, right? One person, did you see him fall down, huh? And then pass from one person to the next person, the next person, the next person. Okay? Very quickly. Is that not true?
Oh my gosh.
Especially you band people, right? Okay, I used to be a man, I understand how it is. So one to the next to the next. So very close-knit groups of people can spread their information very, very quickly. They amplifies, okay? So one causes the next, causes the next. And that's what this is saying right here. Okay, the last one is the signal intermediates. This is insulin receptor. This is what happens again. This is, y'all just got back from Halloween. Your insulin's probably pumping through your body, it's trying to break down all the candy. So again, the insulin's going to be received. Okay, that insulin's then going to start breaking down. It's going to be phosphorylated. That phosphorylate is then going to make an intermediate. That intermediate is glycogen synthetase. So your body's taking that glucose from your sugar, using this intermediate to eventually make glycogen. And glycogen is stored in the liver. Okay? So you've got three main responses. You've got the intermediates, the amplification. What's the last one? Yeah. Oh, the feedback. That's right. So the intermediates, the amplification, and then the feedbacks. Okay? And again, this insulin could be a feedback recognition. This insulin could be then, okay, we've produced enough glycogen. Our glucose level is getting too low, so then that would then cause a feedback to stop the production of insulin.